evidence based on function, evidence based on structure. The evidence based on function includes some key findings related to differences in processing observed in rodents with some echo in the human data. I'll talk a little bit about how the connectivity data, and I'll close with our own findings, as well as those in rodents on gene expression differences along the hippocampal axis. So these are some of the most famous pieces of evidence in favor of some different differentiation or distinction along the hippocampal axis. I'll start with the figures on the right. This is the MRI scan from HM, the famous bilateral MTL injury or resection patient studied for years by Brenda Milner. And one of the classic finding with HM is that he was unable to encode new information. He was able to learn new procedural information, which is what led to the distinction between basal ganglia, type-dependent motor and cerebellar, learning processes, and declarative memory. However, HM did retain some spatial, the ability to encode and retain new spatial memories. This ability was degraded compared to unaffected normals, but he did retain some of it. And so one of the findings you'll see on his MRI scan is that he retains some posterior hippocampal tissue in the posterior aspect of the body and tail of the hippocampus. This led to an early idea, or a persisting idea, that the posterior hippocampus is more densely involved in spatial memory and spatial associative information as compared to the anterior hippocampus. Another piece of evidence in support of this is the famous London cab driver experiment from around 2010, I think. So this is a famous experiment where they took London cab drivers, and one of the aspects of certification to become a London cab driver is to develop a detailed and immediately recallable map of the city of London, like every road, bridge. It's called the knowledge. It's very detailed and difficult to acquire spatial information. So they took these London cab drivers who were in the training phase. They were going through the phase of acquiring the knowledge, and they did MRI scans at different time points along there as they acquired and spent time on this information. They compared the pre and post learning condition brain scans using kind of basic structural MRI. And they found that there was a significant increase in size and metabolic profile in the posterior hippocampus, specifically on the right, although the one on the left was also elevated a little bit. So this is another piece of information in support of the same thing as we saw with HM, that the posterior hippocampus is something that's a little bit different than what's going on in the anterior hippocampus. They didn't see whole hippocampal expansion, even though it's all associative memory that would be theoretically hippocampal dependent. They saw it in the posterior hippocampus. So another piece of the puzzle. So along kind of around the same time as that, the Mosers, of course, the famous Nobel Prize winners from Norway, had developed a theory or a model that distinguishes dorsal versus ventral hippocampal activity in rodents. In rodents, because of the shape of the hippocampus, the distinction is between dorsal and ventral, where dorsal is most analogous to the human posterior hippocampus, whereas ventral is more analogous to the rodents. Or I'm sorry, the rodent ventral hippocampus is more analogous to the human anterior hippocampus. So there are differences between these in rodents. Structurally, one of them is that the ventral hippocampus has a slightly higher proportion of CA1 compared to dentate gyrus and CA3, which has some significance later. That distinction is actually expanded in humans. If you look at just the cross-sectional view of the hippocampus, you'll see much larger proportion of the structure occupied by CA1 as compared to CA3 dentate gyrus that you see in humans. The Mosers further investigated this and looked at, I think it was using tracer studies, I'm not certain, or I don't remember off the top of my head, for connectivity between the two. They saw differences between core and shell connectivity with the nucleus accumbens, which has important implications for reward-based learning. As you would predict, the ventral hippocampus is more densely connected to the amygdala because it's nearby, and which is stands to reason. And based on differences that they had observed, along with these structural connectivity differences, they'd also observed differences in dorsal versus ventral hippocampal activity in fear learning conditions. So conditions that have a component of affective information for the associative memory. That led them to propose that the ventral hippocampus, analogous to the anterior hippocampus in humans, has a specific role in processing for affective information, emotions, things like that. Whereas the dorsal hippocampus is more purely involved in core associative functioning, independent of the affective content of the memory. So that's the Moser model. I have studied hippocampal longitudinal differences for a while. I should follow up on that and say that going back to the Moser model, there's a famous scientist of episodic memory in Toronto, Morris Moscovich, and he has trained many other famous scientists, and he has further investigated this using fMRI and PET scanning, and has observed some similar differences between in humans anterior versus posterior hippocampus, with the anterior hippocampus being implicated in this affective processing. So I, being aware of these different models of hippocampal specialization, and with the opportunity to routinely record from both the anterior and posterior hippocampus in humans, I started investigating this a few years ago. And the first paper, or one of the first ones I published, is the one at the bottom, which is related to differences in the magnitude of theta oscillations. Recall or not, but from my previous lecture, that theta oscillations are a key component of information processing in the hippocampus. These are, let's say, 3 to 9 hertz, depending on your definition, oscillations that are thought to be important for binding together the activity of item-related information. Have been studied extensively in rodents, all kinds of rodent data, but I'm not going to get into it all. But I asked the question whether there were differences during episodic memory encoding, meaning hippocampal-dependent associative memory, episodic memory encoding, between activity that's observed in the anterior and posterior hippocampus. And in that paper, I observed that there was more of this, quote unquote, of this theta activity in the posterior hippocampus. That was important because in some of the first round of papers that had come out of the human cranial physiology world, didn't necessarily see a lot of hippocampal theta, which had been predicted from all the rodent models. For a while, there was a lot of question about is the rodent data related to theta oscillations relevant to humans? My hypothesis was that it might be, but it just might be that the initial studies, which were done, mostly with grid investigations, included an overrepresentation of anterior hippocampal recordings rather than posterior. So I felt like this paper supported the idea that theta exists, because we observe more robust theta oscillations in the posterior hippocampus. But if your data set is sampling more from the anterior hippocampus, the effect won't be as robust, at least during memory encoding. So I moved on from there to start using a different memory paradigm than the free recall paradigm I'd used at the beginning, which was called associative recognition. This, incidentally, is a favorite of the old 1960s. The initial people from Yale that published the descriptive characteristics of human memory. This is the task that they really liked, because they thought it was one of the purest to test the associative memory apart from other things that contribute to memory. One of the big proponents of that and this task in general, Ben Murdoch, just passed away, and he's was one of the giants in memory world for a number of years. Anyway, so I started running the associative recognition task in patients. There's an important distinction where there's a question about differences that you may observe between memory encoding and retrieval. If you're studying memory retrieval, generally speaking, you get cleaner data if you're using a cued paradigm, meaning that there's some sort of cue that shows on the screen that tells the individual what they're supposed to try to remember. In associative recognition, I think there's not it's not like you give in a cue and you have to pick the answer or speak the answer. That would be a paired associates paradigm or something called source memory. In associative recognition, you see both the item and the guy that goes with it. It's paired. You have to make a judgment about whether it's being paired with a new item or with the one that you saw it from at first. Right? It makes a lot of demands on the associative information in order to do the task. And it's been used in fMRI world for a long time. After I started working with Dr. Rugg, who was on the paper, he said, this is the task I like, it's from Ben Murdoch, it's an older task. So I started running that. So the plot on the right is from a paper we published in 2020, looking at both anterior and posterior hippocampal activity in humans doing associative recognition at the time of retrieval, whereas the previous paper that I talked about was looking at encoding-related information. And I thought the effects were fairly robust. The red blotches, this is a standard time frequency plot that I don't need to explain to everyone here. But it's looking at the activity that distinguishes when the individual made the correct association. They recognize that the item was matched up with its appropriate partner, versus in a situation where they recognize that they had seen the word before, but they didn't realize that it had been matched up with a new partner, or was it with their correct partner. So that differentiates, which based on my last lecture, familiarity versus recollection-related activity. And when you look at that contrast, the result is this, these blotches on the right here in time frequency space. And in this case, both anterior and posterior hippocampus exhibit fairly similar effects. If you statistically compare them, they're slightly bigger in the posterior hippocampus. The T statistic maps that we're showing on the left are probably overstate the anterior effect just because there's more electrodes there. But if you look especially at the curves in time, what you'll notice is that if you look at this comparison of associative hit means they got it right, associative miss means they got it wrong. If you look at the plot specifically for these like 3 to 5 hertz theta oscillations, you'll see that the peak this is for anterior hippocampus, it occurs a little later in time than the peak for the posterior hippocampus. We saw something similar in the other paper that I have on here, the theta band paper, during memory encoding. We tested that using something called intratrial phase coherence. And from that, you can measure the offset in the wave. How many degrees is the offset between the wave observed at the same time at location A versus location B? The offset ends up being about 40 degrees, which for an average 4 hertz oscillation means that there is a traveling wave moving along the dorsal to ventral hippocampus at a speed of about 1.1 meters per second. And this idea of traveling waves is becoming kind of hotter and more interesting. Having only 2 points in space to do the analysis isn't ideal for a traveling wave analysis, but there's a lot of interest in how these traveling waves are working, where they're coming from, etc. If you take a very relatively simplistic model that theta oscillations are generated in the septum, cholinergic, which is one of the models for how they're generated, then a dorsal ventral posterior to anterior in humans propagation makes sense. So moving on from there. So the other thing that I wanted to bring up, this is the follow-up paper that we did to that one, is this idea of pattern separation versus pattern completion. Thinking back to my last lecture, one of the things that I wanted to emphasize is that the function of the hippocampus is to form novel associations. At the time of retrieval, there's 2 things that it has to do in order for you to recollect a memory. It's thought that there's a 2-part process. One is pattern separation. So if you imagine that you have I don't know, let's make it easy. Let's say there's 100 neurons that are representing a memory. The hippocampus is not, all the details of the memory are not stored in the hippocampus. The job of the hippocampus is to put together the right combination of these, simplistically determined 100 neurons that are characteristic of the memory. But at any time in the brain, there's thousands of neurons firing. So what the hippocampus is thought to be doing is to first separate relevant patterns from non-relevant patterns, and then to link together whatever components of the pattern from memory have not yet been activated. So these 2 parallel processes, pattern separation and pattern completion, are going on all the time when you form a memory. There's some controversy about that. I don't want to say that's not like settled Newtonian law. It's just sort of one of the kind of leading models for what the hippocampus is doing. Now, pattern separation, based upon some very basic neurophysiology that I honestly can't rehearse off the top of my head, is thought to be more predominantly governed by CA1. whereas pattern completion is thought to be more predominantly governed by CA3 dentate gyrus. Has to do with recurrent connections, some other properties of the neurons themselves. As I said, I don't want to get over my skis and trying to get into the weeds on it. But that basic idea that there's a difference in pattern separation and pattern completion between CA1 and CA3 dentate gyrus is relevant because, as I said already, CA3 dentate gyrus is relatively overrepresented in the posterior hippocampus compared to anterior. So that would lead you to predict that there should be some kind of difference in at the time of retrieval when this pattern separation, pattern completion process is going on. So these gamma oscillations are, along with theta, are relevant and interesting. And we were testing whether there was a difference in the relative activation of these gamma oscillations, which are higher frequency brainwaves, basically, between encoding and retrieval. So what this plot is showing is if you take a ratio, essentially, between encoding, the magnitude of activity in encoding, and the magnitude of activity at retrieval, what's the ratio? And this is using free recall, so it's not the perfect paradigm for looking at memory retrieval, but since we're comparing anterior to posterior hippocampus, both structures are seeing the same paradigm. And we saw a pretty stark difference between this ratio with greater posterior related activation at the time of retrieval. So, and we interpreted this as maybe being consistent with this higher pattern completion role that's occurred, where pattern completion is more prominent at the time of retrieval and free recall. There's another finding that we didn't see that I could get into related to some findings from Laura Colvin at UT Austin. So that's a quick intro to some of the physiological evidence that I published related to anterior versus posterior differences and kind of putting it in context. So now let's put it into the broader context of other models. So this is a well-cited 2013 paper from Jordan Poppink, who was a student of Moskowitz in Toronto. And this kind of summarizes all of the observed connectivity differences that exist between the anterior and posterior hippocampus. I'm not going to just read the slide, but basically there's a lot of differences as you would predict, right? And the anterior hippocampus is more densely connected to temporal pole, ventral temporal cortex. It's obviously more densely connected to the amygdala. There's this nucleus accumbens, core, shell distinction, which is related to reward processing. All of these things just support the general idea that these areas are different. My view, and I think probably the prevailing view, is that an affective versus non-affective distinction between the structures does not capture the full complexity. I mean, that's certainly supported in my data. You see differences in anterior versus posterior hippocampus on a task that has no particular manipulation of the emotive content of the words, then I don't think you can conclude that a model based on affective processing is capturing the whole story. Certainly that could be part of it, probably is, but there's more to that than what's going on. So Poppink proposed a different model. So Poppink's model is that there is a difference, there is a gradient of information along the anterior to posterior hippocampus. The anterior hippocampus is involved in what's called gist memory. Yeah, I kind of remember it. I remember the category. I remember it was in, a mammal or like a horse-like mammal, but not the detail. The detail, according to Poppink's model, is represented in the posterior hippocampus. So that would be where the full detailed memory. Oh, it's a zebra with whatever, missing an ear or something like that. That's where the posterior hippocampus would be necessary to capture that level of detail. This is called the gist detail model, and it's probably one of the leading ones that's out there. So that's what the tag on the slide is referring to as representational gain. How much detail is included in this memory? The anterior hippocampus is necessary for capturing gist information versus detail. Now, you might think that would lead to a two-part process for retrieval, where first you recover the gist and then you recover the detail. That's really not clear. I don't think Poppink is that there's no real evidence for that. And in fact, my own data would say that, well, based on the timing of when the theta oscillation is going through in this traveling wave model, it doesn't really fit that way. And so if there is this representational gain, just detail difference, it wouldn't support the idea that first you recover the gist. And I don't think that that's true. But interesting. Obviously, anytime you have a model, you have physiological measures, and you have specific predictions that would evolve from that model, i.e. you can manipulate the level of gist or detailed information. You can ask somebody if they're recalling, do you remember a vacuum? Was it a household item or an outdoor item? Was it a vacuum cleaner or was it a dustpan? That you can experimentally manipulate those two things, right? So we could test the gist detail model. And in fact, I proposed this to the NIH over a couple of grant cycles and was never funded. But it's out there, and maybe one day we'll have the funding to run this experiment. So this is really the core of the gist detail model. It's the key rodent finding that led to this idea of representational gain. It's really, really nice and subtle. So as you recall from my previous lecture, there are these things called place cells, right? So a rat and a human, a little bit, is running through some kind of spatial environment. And there's cells that fire specifically and reproducibly at the same location in that spatial environment. This is the core of how the hippocampus is representing space, right? So that's what the different colors of dots are representing on the slide of this little figure on the right. That there's the yellow dots represent place cell number 2, it always fires there. And the green dots represent place cell number 4, always fires there. Now these are flexible. It's not like they have to fire only at that one location in space for eternity. It depends on the environment. There's the ability to remap, etc. But this is what the Mosers, won the Nobel Prize for. This is pretty well accepted and understood. If you look at how big is the place field, well, what's a place field? Well, a place field is just asking what space in my maze does this place cell fire over? Is it 2 centimeters of space? Is it a meter of space? How wide? What is the selectivity of my place cell? That's the place field, right? If you look at how wide the place fields are in a rat, this is pretty reproducible, or it's been, I guess, pretty well accepted. The place fields in the ventral, analogous to human anterior hippocampus, are wider than the place fields in the posterior hippocampus, right? You can see how that, along with some other evidence, motivated this popping model, the gist detail representational gain model. The anterior hippocampus looks more broad, approximately in the back corner of the room. Posterior hippocampus, dorsal in rodents, is more specific. Well, I'm at this more precise location right near the door, right? And so the idea is that both of these types of information involved representing the spatial location, gist detail, right? So that is this is really the crux of it. This is the main piece of evidence for it. All the other stuff, the connectivity, you can make a connectivity map look kind of how you want it. But this is really the core physiology that's at the heart of gist detail. So, I think I told you in the previous lecture that, humans, I don't think care that much about spatial information. This is certainly not in the way that rodents do. We care a lot about time. We care a lot about when something happened. So that, of course, motivated our attempt to go find this analogous population of place cells called time cells. I don't want to get into it. Basically, time cells function like place cells, but in the temporal domain rather than the spatial domain, right? And so when we went looking for time cells and found time cells, you can see we had this very obvious hypothesis. The time cells in the posterior hippocampus will mark out a broader time field than the time cells in the anterior, a narrower time field than the time cells in the anterior hippocampus. If time cells and place cells are the same, then they should do something like that. And we didn't really see it. It doesn't quite fit. So that's science, right? So at that point, we're working on all these problems that, as you guys know, I collaborate with Janet Kanapka, who does gene expression profiling, and this was her postdoc at the time. Stefani's now has his own lab in South Carolina. And we were working on gene expression analysis from the collateral temporal cortex related to brain oscillations. That was great. But we were talking at one of our lab hangouts about what we could do with the hippocampal data. And I said, well, I'm working on anterior posterior hippocampal differentiation. I just read this review paper by Banzelo and Dong. And one of the big pieces of evidence they cite is that there is a gene expression gradient from the dorsal to ventral hippocampus. This is in mice, not in rats. That there are clear genes that seem to be represented anterior versus posterior. If I'm being completely honest, the gene expression data was really structured as head, body, tail. So 3 parts, not 2 parts. And that's maybe better. I've been simplifying it to say anterior versus posterior because we just don't get that much, as much detailed representation with electrodes. So I've been calling it anterior posterior. But one of the things we acknowledge in all the papers is to say that this may miss some of the subtlety between head, body, tail. So I should acknowledge it. So these differences in gene expression are done using kind of standard polygenetic techniques, looking at how families of genes are being expressed together. It was pretty well understood, and I said, well, we can do this study in the human tissues that we get that produce from temporal lobectomy operations. This is, I think everyone in the room has done a temporal lobectomy now at this point. Maybe not right. As you know, we spend a lot of time, we make the section smaller, we take the hippocampus off its vascular pedicle, we do it in a way that tries to preserve as big and long a specimen as possible. Part of it is just that it's kind of nice to do that. And then second is that, well, if you do that, then you can differentiate stuff that's going on in the anterior posterior from hippocampus from the posterior with some degree of confidence. So we did that for this in the paper that I'm showing here. But we knew that we would face some hurdles. These are epilepsy patients, XYZ. So we had to wait till we got enough specimens where we had a full 3.5 to 4 centimeter hippocampal specimen. The patient didn't have MTS on pathology or some other lesion. They had not had intracranial electrodes because we didn't want the gene expression to be affected by scarring or whatever from the electrode. So we had to wait till we collected enough patients. So it took a long time till we were able to winnow down the sample to ones that we felt would inspire the most confidence. But we did the same analysis using gene expression profiling. And this is using something called single-cell RNA sequencing. So single-cell RNA sequencing uses special tools or special machines to break up the tissue and process gene expression data from individual cells at a time. And because you can characterize what type of cell it is based on the gene expression, you can assign different genes to having been expressed in different cell populations. Astrocytes, this is not novel. I mean, it's been around for a while, but that's the reason that on the x-axis here, it has different cell types, CA1, CA2, granule cells, interneurons, etc. Right? And then on the y-axis are groups of genes that are associated from a bunch of studies, GWAS, all the accumulated knowledge of human genetics with these different traits or disease states. And so we're able to say, well, here's these gene groups, gene families of expression. These are the cells they're expressed in, and we can also look to see which ones are differentially expressed in the anterior versus posterior hippocampus. So cataloging these DEGs or differentially expressed genes was kind of the main focus of the paper. Broadly speaking, about a third of the genes that were identified as being differentially expressed in mice were also differentially expressed in humans. Some genes just aren't persistent in humans, some genes just weren't differentially expressed. But there was enough there, and certainly more than by chance, that we felt like there was a good proof of that what we were doing was capturing some of the same information as the big time mouse papers. But the thing that you'll see also is that conditions such as schizophrenia, major depressive disorder, bipolar disorder are more strongly found in anterior hippocampal locations, and especially for granule cells and interneurons. Whereas genes associated that are linked with cognitive function and intelligence are more strongly linked with posterior hippocampus. And then ASD, autism spectrum genes, of which there are many, are more strongly found in the posterior hippocampus. That's really the most fascinating thing. So kind of to summarize, this is, I kind of walked through, in this paper, we, with the help of our previous medical illustrator, made this really nice looking cover art, which made the cover. So just to summarize, last slide's pretty, I've tried to show you electrophysiology. I haven't really gone into a lot of the connectivity data other than quoting it, but electrophysiological and gene expression evidence in humans that anterior and posterior hippocampus have distinct function. And it's something that I continue to study with all the analyses that I run for human memory. And I should take questions.
Amazing. It sounds like the tasks are an important selection process, and that it can affect what kind of activation you get.
Yeah, that is absolutely right. There are a lot of tasks out there. And it's not just, there's like the task, broadly speaking, there's individual manipulations. Do you represent the word? Are the words you're presenting, are they semantically related? Is it just random? Most of the time we just use ones that have like a baseline level of at chance semantic relationship, but you can manipulate that. And if you manipulate the semantic relationship, you can look at how that affects anterior posterior distinctions. There's a separate model, which I didn't get into here, called the PMAT model, posterior medial anterior temporal model. It sort of builds on gist detail, but it doesn't say that. You know what I'm saying? It's more of a cortical model. It's not related to hippocampal specialization, but it claims that there is a, broadly speaking, distinction in brain networks between these posterior medial network, which we'll leave the hippocampus out, includes perihippocampal cortex, posterior cingulate, the midline anterior regions, not the anterior cingulate, dorsal to that, angular gyrus. That is thought to, it's a lot of that overlaps with the quote unquote default mode network, but it's thought to be involved in memory retrieval, spatial processing, spatial memory, spatial memory integration, versus the anterior temporal network, which is involved more in semantic memory and semantic detail. But if you wanted to test that, you would have to do something like a manipulation of the semantic content of the items versus maybe the spatial content. Where on the screen did the word appear? Stuff like that. But I mean, like anything in the world, and the more tasks you think about, the more you have kind of at your disposal to use if it fits with the question that you want.
Brad, clinically speaking, obviously we talked about dominant versus non-dominant hemisphere. And the differentiation that you show between, so are there physiological differences between dominant and non-dominant hemisphere, and are there genetic expression differences between dominant and non-dominant?
There are no, so this is the whole cortex. There have been no studies, it's been looked at a lot, that show convincing gene expression differences between dominant and non-dominant hemisphere, including dominant and non-dominant hemispheres. Jenna Kanopka says that every time she gets a medical student in the lab, they always want to run that experiment, she said, and she said she's going to get a bell to ring every time someone asks them, because everybody assumes there are, and there just aren't, which is shocking. I would have definitely said that if I joined her lab, I would have done the same thing. Oh, what about dominant? Yeah. It just for whatever reason, whatever the differences between the hemispheres is not. There's not a clear reflection of that in gene expression.
So what about the physiological activity?
So physiologically, if you are looking just at the associative information, remembering let's say that you have 10 associative tasks, like that you have a spatial associative task, you have a verbal task, you have something else that you use animal pictures and you use pictures of mountains or something like that. If you look at just the whatever contrast, pick out the associative information, that is pretty clearly symmetrically represented in the hippocampus. Differences in left versus right activation come on more so if you look at the cortex, obviously. If it's a verbal memory task, you'll see more dominant hemisphere activation. If you have a purely spatial task, there's no words involved whatsoever. You'll see potentially more right-sided, like neocortical activity. But my takeaway from all these years of studying it is that for the core question of the association, the thing that the hippocampus is supposed to be providing, they're really even between, there's not a difference between the two hemispheres. So when I counsel patients about the impact of whatever intervention we're doing, like laser ablation or something, or lobectomy, I always differentiate the verbal ability, using a word in its conventional use, not a memory-related ability, from the episodic memory component, the memory ability. And the memory ability is impacted essentially equally by dominant, non-dominant interventions, whereas obviously the verbal ability is different. But the corollary, which I assume everybody cares about, and which I'm curious about, is, well, does that mean that we should be tailoring our hippocampal ablations or resections based on localization of seizures plus concern for residual cognitive function? If it's clear that the anterior hippocampus is more active, interically or ically, does that mean we should spare the tail to help preserve some of this associative function? And the answer is maybe. I don't know. That experiment may be ongoing by accident, because laser ablation usually people are kind of prioritizing the anterior hippocampus a little bit more so now with laser, rightly or wrongly. I will say that I've never been committed enough to say, well, we should just spare the posterior, except in very one case, over the years. So I thought, good idea. Generally speaking, you'll see more seizure activity in the anterior. I don't know if it's just because it's bigger, so it generates bigger intericals. The electrodes are kind of more right in the heart of it more often. I don't know. But there are a handful of cases where you'll see the seizures really starting posteriorly. Those are the minority. But it's a
Regarding the time, why do you think that model didn't work? Is it data acquisition? Is it hard to model? Or is it more complicated?
There's a few options. One of them is that the model is wrong. The second option is that, or the model is maybe right for spatial information, but not for, which is different. Another option is we just didn't sample enough time cells. If we had sampled, enough time cells, we would see more of them. And that's probably the more likely answer, is that we just didn't see enough evidence consistent with it, but doesn't mean it's not there. Having said that, I didn't get into a related class of temporally sensitive cells called ramping cells, which cooperate with time cells. They're supposed to be kind of like grid cells, like there's grid cells and place cells. The analogy is that there's ramping cells and time cells. We did see more ramping cells in the entorhinal cortex, which is the same thing you predict from other spatial models where entorhinal provides grid-like activity. We did see that, but not the anterior posterior distinction for time cells.
Thank you.
Good. I could talk about this all day, so we should stop here before I go.